Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with Shatrin Sawadi. And today I'm here to discuss about the primers and primase in detail. I got request to deliver the lecture about the primers used in the replication mechanism by the cell and the primers which are used in the PCR. So in this lecture I will discuss the primers in DNA replication use and naturally synthesized in the cell, and the primase which are responsible for the formation of the primers. So I will discuss how do primers work in the DNA replication and the primase structure in detail. In primase structure, I will share the knowledge about the bacterial primases, archaeal and eukaryotic primases, and in eukaryotic primases, I will discuss in detail about the human primase. So now I'm starting from the primers in DNA replication. What are basically the primers? Primers are small pieces of the RNA. They're about 5 to 15 nucleotide long. And the primers which are used in the PCR for the amplification process are small molecules of the DNA. I will discuss in detail about the primers used in the PCR. So the primers are the small pieces of RNA about 5 to 15 nucleotide long. Primers are required for DNA synthesis because no known DNA polymerase is able to initiate polynucleotide synthesis, mean the DNA synthesis or DNA replication. DNA polymerases are 1, 2, and 3. And the polymerase 3 is responsible for the initiation of the DNA replication. So the polymerase about the DNA polymerase, I will deliver the lecture very soon about different types of the DNA polymerase. So must keep it in your mind, the DNA polymerase do not work, do not initiate the DNA replication without the primers. And these primers are synthesized by the special types of the enzymes. And these enzymes are known as primases. Now, what are primases? Primases are special RNA polymerase. Primase synthesize a short piece of RNA, means the primer, that is complementary to the template DNA. Must keep it in your mind. That small piece of the RNA, which is primer, is complementary to the template DNA strand and forms hydrogen bond with it. How it bind with the single-stranded DNA with the help of the hydrogen bond. This gives DNA polymerase the starting point it needs to initiate synthesis or replication during DNA replication. Is it clear? So what is primase? Primase is just a RNA polymerase enzyme which is responsible for the formation of the short pieces of the RNA that are primers. And these primers are responsible for the DNA replication because DNA polymerase do not work without the primers. Okay. Now, how do primers work in the DNA replication? Special proteins are responsible for loading primases at the origin of replication so that the leading strand DNA synthesis can commence. Now, I will show you here. This is the DNA replication mechanism in which the DNA, double-strand DNA converted into the single strand, and what will happen that the primases are loaded by the special types of the protein which are present in the nucleus of the cell. So, what will happen? Now, in a subsequent step, other replication protein causes primase to initiate DNA replication on opposite lagging strand. As we are familiar that two strands are formed after the unwinding of the DNA. One is the leading strand and one is the lagging strand. Leading strand synthesize the DNA from 3 to 5 in the direction of the replication fork, but the lagging strand form the, or you can say that start the replication from 3 to 5 n, but opposite to the replication fork. So the replication at the leading strand is continuous, but at the lagging strand this is discontinues and produces the small fragments along the parental strand in the form of and these fragments are known as the Okazaki fragments. Is it clear? Okay. Now these 2 points are clear. After both the leading and lagging strand, primers have been elongated by the DNA polymerases. The RNA primers are enzymatically. Now what will happen when the replication complete? Then the RNA primers are enzymatically eliminated, and the resulting gap in the DNA sequence is filled by the DNA polymerase 1 and the DNA ligase. Is it clear? So what will happen? First of all, the primases attached with the single-stranded DNA, and then produces the primers, and these primers attach with the single-stranded DNA with the help of the hydrogen bond, and then start the replication. Then what will happen? These primers remove from these 2 strands after the completion of the replication by the help of the enzymes. Is it clear? Okay. Now, there's some interesting information for you. That single-stranded DNA in phage M13 genome, phage, this is bacteriophage M13, utilizes RNA polymerase instead of primase to initiate the DNA synthesis. Must keep it in your mind. And in that case, like the mitochondrial DNA replication, in mitochondrial DNA replication, initiation is that the mitochondrial RNA polymerase synthesize a polymer that is not displaced from the template. What will happen in this case in DNA replication? What will happen? The mitochondrial RNA polymerase synthesizes the polymer, and that polymer attached with the template DNA. Then what will happen? Then the special RNAs MRP cleave the ribopolymers at specific site, enabling the exposed 3-hydroxyl terminus to serve as a primer for DNA replication. So this is the difference between the eukaryotic and prokaryotic types of replication, and the mitochondrial DNA replication, as well as there is some information about the phage M13 DNA replication. Is it clear? Okay. Now, primases structure. I'm going to discuss about the bacterial as well as archaeal or eukaryotic primases structure. So first of all, I'm going to discuss about the bacterial primases. Bacterial primases are monomers and consisting of 3 domains. How many domains? 3 domains. One domain is the N-terminus domain. I will show you here. You can see here there is an N-terminus domain, and N-terminus zinc binding domain is known as ZBD, and you can see here this is the ZBD. Why it is called ZBD? Because the zinc is present here, so zinc binding domain. Other one is the RNA polymerase domain. Other is the RBD, means the RNA polymerase domain, and the C-terminus, which is the helicase binding domain, HBD. Is it clear? So this is the helicase which attached with this primase. So what will happen? Now this N-terminus domain has a zinc finger motif and is likely responsible for the initiation specificity of this enzyme. Is it clear? Now the second one point is that the central catalytic domain binds single-stranded DNA and catalyzed RNA polymer initiation and elongation complementary to it. Is it clear? This start initiation, the initiation and then elongation of the RNA primer. The C-terminal domain interact with the other proteins, including DNA B helicase. So C-terminal is responsible for the attachment of the primase to the helicase, and the primase is represented by DNA G in prokaryotes, and the helicase is represented by DNA B. Is it clear? DNA G and DNA B. Now you can see here this is the DNA B, which is the helicase, and this is the DNA G, which is the primase. So both form the complex for the for its activity. Okay. In Escherichia coli, the primase DNA GB and helicase DNA B interact to form a complex. As I have shown you here, this is the complex. Okay. This interaction stimulates both the primase and helicase activities and relaxes the sequences specificity of the DNA G, allowing DNA G to successfully prime Okazaki fragment synthesis during lagging strand synthesis. 3 DNA G primase proteins associating with each DNA B helicase to form the primosome. Now DNA GB and DNA G and DNA B form the structure which is known as primosome, which I have shown you here. This is the primosome structure, complex structure. Okay. E. coli DNA G has a molecular weight of 60 kilodalton and contains 581 amino acids. So it is clear upon you people that in the case of the bacteria, what will happen? The primase form the complex with the helicase for its activity, and this complex is known as primosome. Okay. Now I'm going to discuss about the eukaryotic primase. Eukaryotic primase consists of small primase subunit. Now the primase in the prokaryote consists of 3 subunits, 3 parts. Now here the primase consists of the 4 parts. Number 1, small primase subunit. Number 2, a large primase subunit, which are the actual part. And the third one is the regulatory phosphoprotein. And the fourth one is the DNA polymerase alpha. The small subunit has a primer synthesis activity that is modulated by the other 3 proteins in the complex. Now you can see here, this is the eukaryotic primase structure, and this is from the eukaryotic, this is the human primase structure, and you can see here the 4 parts. This is the small subunit, this is the large, this is the phosphoprotein, and this is the alpha polymerase. Okay. Now human primase structure I'm going to share with you. This is simple eukaryotic, but I will share something interesting about that. The primer active site for RNA primers synthesis resides in the primary S subunit. Is it clear? Means S subunit means small subunit. So where the RNA primer synthesize in this small subunit. Okay. Now the second one is the large subunit. It's important for the regulation of primase activity and primer transfer to the polymerase alpha. Is it clear? Okay, that is very interesting. Okay, you can see here the comparison between the bacterial and the human primase, and this is clear that the bacterial primase consists of the 3 parts, and the eukaryotic means the human primase consists of the 4. Is it clear? Okay. Now you can see the human primase structure. This is the secondary structure element that make up the prime folds are shown here, and you can note down this alpha sheets and beta sheets. Are you familiar with the protein amino acids chains, alpha chain, beta chains in the protein? This is that. Okay. Now, due to the discontinuous nature of the DNA replication, primase activity on the lagging strand is required throughout the replication process. Must keep it in your mind. Because of the discontinuous DNA replication on the lagging strand in the form of the Okazaki fragments, required primase activity throughout the replication process. In eukaryotic cell, the presence of the primase at the replication fork is secured by its physical association with the DNA polymerase alpha. As you have seen here that the polymerase alpha is attached with the primase, and this polymerase alpha secure that primase. Is it clear? So association with the DNA polymerase F5, which extends the RNA primer with deoxynucleotide. I hope so the lecture will be clear upon you people. If you still have any question, write in the comment box. I will try my best, and I will be back with the primers and the PCR soon, inshallah. Till then, Allah Hafiz.